Dear students, I would like to start today with the skeletal muscle lectures. The skeletal muscle lectures include 3 lectures, 23, 24 and 25. We will start today with the first one, which is related to the general principles of the muscle function, and also we have to finish with the shape of the skeletal muscles and types of proteins located in skeletal muscles. Moving to the skeletal muscle tissue, and in comparison, what is the shape of the cardiac muscle and smooth muscles? That is the shape of the skeletal muscle. They are appearing striated, and if you look to the shape of the skeletal muscles are appearing striated, and they have something which is called alternating dark and light bands. They are attached to the skeleton or to the bone, and that is helpful for the production of their action, which is locomotion. The second type of muscles that is located in the heart. It is the cardiac muscle, and if you look to the shape of the cardiac muscle, they are also appearing striated. They have something which is called the alternation between dark and light bands, with the presence of another two criteria. Number one is the branching of the cardiac muscle fiber, also the presence of the intercalated discs that connect the muscle fiber together. If we go to the last type of the muscle, is the smooth muscles, and the smooth muscles are small in size. They are spindle shaped, and as you see, they have no striations. And they are interconnected by the gap junction. They are interconnected by the gap junction. So we have skeletal muscle. They are striated, alternating and dark bands, cardiac muscle, and smooth muscles. Striated in shape, cardiac muscle, smooth muscles, non-striated, and they are connected by gap junction. If we go to the skeletal muscle tissue, so named because most skeletal muscle fiber move bones, they assist mainly in locomotion. Skeletal muscle tissue is striated. As we mentioned, we have an alternating dark and light bands. They are supplied by the somatic nervous system, so they are under the voluntary control. Some skeletal muscle work or controlled subconsciously to some extent, like the muscles of respiration that are controlled by the respiratory centers located in the medulla oblongata. Cardiac muscle, found in the wall of the heart. It is striated in shape like the skeletal muscle. Its action is involuntary, and as you remember, its contraction and relaxation is usually controlled by what? By the impulses coming from the pacemaker of the heart. Smooth muscle, located in the wall of hollow viscera, like gastrointestinal tract, like the uterus, like the ureter, like the airways, like the blood vessels. They are not striated, no alternating light and dark bands, controlled by the autonomic nervous system, so they are usually involuntary. So, moving to the properties of the muscle tissues. Number one, excitability. What's the meaning of excitability? Excitability means ability to respond to a stimulus. If you stimulate the muscle tissue, they are going to respond, and their response is usually appear in the form of electrical event and mechanical event. And the mechanical event is the contractility, which is the ability to produce the contraction and relaxation. So the process of contractility means the ability of the muscle to transfer the electrical and chemical energy into mechanical energy. Extensibility means the ability of the muscle to stretch without being damaged. The ability of the muscle to stretch without being damaged. Last, we have the elasticity, and elasticity means the ability to return to the original length. The ability to return to the original length. That is the elasticity. So the muscle tissue has 4 main properties, including excitability, contractility, extensibility, and elasticity. Regarding to the general functions of the muscle, skeletal muscles are helpful in locomotion, in maintenance of posture, production of respiration, and communication, as well as thermogenesis. These are general criteria or general function for the skeletal muscle. Skeletal muscles are helpful in the production of voluntary locomotion, maintenance of posture, respiration, communication, and production of heat by thermogenesis. While smooth muscles produce peristalsis of the intestinal tract, also vasoconstriction of the blood vessels to maintain the peripheral resistance and maintain the blood pressure. So they have function which is constriction of organs and vessels. Heart producing its action, which is the production of cardiac output by heart pumping. So that is the general function of the muscle tissue. If we look to the structure of the skeletal muscles are inserted into the bony part by the tendons, and the tendons is a site of attachment or site of insertion of and origin of insertion of the skeletal muscle to bones. If we would like to have a cross section in the skeletal muscle and look to the structure of the muscle as a whole, we can draw it like this. That is the surrounding of the skeletal muscle from outside, and this part is known as epimysium. Epimysium. And if we go and look to the right, we will find that we have something which is called the muscle fascicles. The muscle fascicles. And these muscle fascicles are separated from each other by something called perimysium. They are covered by something called perimysium. So we have epimysium and we have perimysium. If we go and right, that is the muscle fascicle, and each muscle fascicle is covered by the perimysium, and the whole muscle fascicles are covered from outside by the epimysium. If we look to the muscle fascicle is formed of something called the muscle fibers. And if we go to the muscle fiber, which are collected together to form the muscle fascicles, they are formed of something inside which is called the myofibril. So the outer part of the skeletal muscle is covered by the epimysium. The muscle is formed of different muscle fascicles. The muscle fascicles are covered by perimysium. Inside the muscle fascicles is formed of the muscle fibers. The muscle fibers are separated or covered from outside by something called endomysium. And the muscle fiber is formed from something which is called the myofibril. So again, that is the cross section of the skeletal muscle. Epimysium covering the muscle from outside. Muscle is formed of multiple muscle fascicles, and these muscle fascicles are, for each muscle fascicle is covered by something called perimysium, and the muscle fibers is containing the muscle fibers, and the muscle fibers are covered by the endomysium, and each muscle fiber is containing something which is called myofibril. So we can conclude that the myofibril is the structural unit of the skeletal muscle. The structural unit of the skeletal muscle. So to look again to what we see, that is the perimysium, and this, that is the periosteum of the bone, that is the periosteum of the bone, and that is the tendon insertion. And if we go and look to the muscle, we have here the epimysium covering the muscle from outside. Then we have the muscle fascicles, and the muscle fascicles is containing the muscle fibers, and the muscle fibers is containing the myofibrils. Microscopic anatomy of skeletal muscle fiber. We have some points we would like to cover. What is the meaning of the sarcolemma? Sarcolemma is the outer plasma membrane of the muscle fiber. We have something we have to know clearly. What's the meaning of the T-tubules, and what's the meaning of the sarcoplasmic reticulum, and what's the meaning of triad. But before, we have something which is called the cytoplasm of the muscle fiber, and this is called the sarcoplasm. And this sarcoplasm contains the myofibrils and containing the sarcoplasmic reticulum. So we know that we have something which is called the sarcolemma. Then the sarcolemma has invagination called the T-tubules, and we have the sarcoplasm, which is the cytoplasm of the muscle fiber that contains the myofibrils, and it also contains the sarcoplasmic reticulum. We have that is the sarcolemma. Sarcolemma has something which is called invagination to form the T-tubules or the transverse tubules. So we have something which is called the T-tubules or the transverse tubules. These T-tubules are invagination from the sarcolemma deep into the muscle fiber. So that is the sarcolemma, which is the outer covering of the muscle fiber. It has something which is called invagination to form the T-tubules. And if we look near the T-tubules, we have the type of tubules which is called longitudinal tubes, which is the sarcoplasmic reticulum, and this sarcoplasmic reticulum has dilated ends called the cisternae of sarcoplasmic reticulum. That is the endoplasmic reticulum of the skeletal muscle, or the sarcoplasmic reticulum. And this sarcoplasmic reticulum is containing calcium in a combination with a protein called calciquestrine. So calcium is stored in the sarcoplasmic reticulum, binding with a protein called calciquestrine. Looking to the three structures, the T-tubules, the sarcoplasmic reticulum on both sides, they giving us the term triad, giving us the triad. So what is the triad? It includes the T-tubules and the two dilated ends, or the two cisterns or cisternae of the sarcoplasmic reticulum. So again, we have the sarcolemma, which is the plasma membrane covering the muscle fiber. The T-tubules are invagination of the sarcolemma deep into the muscle fiber. Sarcoplasm is the cytoplasm of the muscle fiber, and it contains myofibrils, which are little threads that contains the contractile proteins. And we have the sarcoplasmic reticulum that encircle each myofibril. It has a dilated ends, which is the terminal cisterns from both sides. Sarcoplasmic reticulum stores calcium and release to initiate contraction. If we go to the skeletal muscle sarcoplasmic reticulum system, it is elaborate, smooth endoplasmic reticulum, runs longitudinally and surround each myofibril, form chambers called terminal cisternae on either side of the T-tubule. A single T-tubule has two terminal cisternae to form a triad. Sarcoplasmic reticulum stores calcium when muscle not contracting or in the relaxation state. Contain large amount of calcium bound to a protein called calciquestrine. When stimulated, calcium released into the sarcoplasm. Sarcoplasmic reticulum membrane has a calcium pump that functions to pump calcium out of the sarcoplasm or from the cytoplasm back to the sarcoplasmic reticulum after contraction. So we have to know that the calcium is released to initiate the process of contraction, and we pump the calcium again to the sarcoplasmic reticulum by active transport mechanism to terminate contraction and start relaxation. Calcium is stored in the cisterns of the sarcoplasmic reticulum in a combination which is known as in a combination with a protein called calsequestrin. Going to the sarcomere. Sarcomere is the structural and functional unit of the myofibril. So the myofibril is formed of the sarcomeres. And if we look to the structure of the sarcomere is the part of the myofibril that's present in between two Z lines. So that's one Z line and the other Z line. And the sarcomere is containing two types of bands, or is containing two types of we contain thick filaments. It contains thick filaments in the center. So you can draw the Z line and Z line, and in the center we have the thick filament, and this thick filament usually contains the protein which is known as myosin. And this protein, which is myosin, is characterized by the presence of a tail that's present in the thick filaments. While it has something which is called the head, which is coming out from the thick filaments, which contain also a head, and that head of myosin filaments are coming out from the filaments. So this is known as the thick filaments. Attached to the Z line, we have another type of filaments called thin filaments. So that is the thin filaments, and these thin filaments are attached to the Z line. These thin filaments containing three types of protein. It contains actin protein, troponin, and tropomyosin. It contains troponin and tropomyosin. So it contains the actin protein, troponin, and tropomyosin. That is the sarcomere, present in between two Z lines, containing thick filament in the center, containing protein myosin. Protein myosin has something called head myosin coming out. We have thin filament attached to the Z line, and they contain three types of protein: actin, troponin, and tropomyosin. If we go and look, we have part of the sarcomere in which we have overlap between thick and thin filaments. We have here thick filaments and thin filament, thick filament, thin filaments. So this part of the sarcomere contain overlap between thick filament and thin filament. We name this as a band. So what is a band? It is a part of the sarcomere in which we have something which is called overlapping between thick and thin filaments. We have also on the periphery of the A band part of the sarcomere in which there is only thin filaments. This is known as I band. So the I band is a part of the sarcomere which appear lighter. This is the part of the sarcomere A band appear dark. That's the part of the sarcomere which appear light, which is called the I band. The I band is a part of the sarcomere which contains only thin filaments. Going to the A band, we have part of the A band contains only thick filaments. Contain only thick filament. It's called H zone. So what is the H zone? It is a part of the A band in which there is only thick filament, no thin filament. H zone present in the center of the A band contains only what? Contains only thick filaments. And in the center of the sarcomere we have a line, we name it as M line. M line. So that is the sarcomere. In the center we have a line known as the M line. So again, what is the shape of the sarcomere present between two Z lines? Contains in the center thick filaments formed of a protein called myosin. In the periphery we have thin filaments. Thin filaments in the periphery, and this thin filament in the periphery containing actin, troponin and tropomyosin. A band is a part of the sarcomere that contains thick filaments overlapping over thin filaments. I band contains thin filaments only. H zone containing thick filaments only. And in the center we have the M line. That is thick filament. Thick, thick, thick. And we have here on the periphery thin filaments. Looking for the sarcomere. Sarcomere are found within the myofibrils. They are arranged beside each other, and they are present in between two Z lines containing thin filaments in the periphery that's mainly formed of actin and troponin, and we have tropomyosin. Thick filament that contains mainly the myosin, and the head is coming out to interact with actin during muscle contraction. The discs separates one sarcomere from the next. A band is a part in the contains the dark part, and this containing actin filaments overlapping over myosin filament, or thick filaments overlapping the thin filaments. We are now moving to the proteins in the muscle. We have the muscle proteins, and we classify the muscle proteins into contractile proteins, regulatory proteins, and structural protein. Contractile protein including mainly the myosin, and this myosin makes the thick filament. It is present in the thick filament. It contains part which is called the tail and part is called the head. The head is coming out from the thick filament, and its function is to interact with the protein which is the actin during the skeletal muscle contraction. This head of myosin has the capacity to hydrolyze ATP and release energy from the hydrolysis of ATP. Then we have actin. Actin makes the thin filaments, and it is arranged in the form of double helix. It's a globular protein formed of double helix. And if we look to the actin has something which is called binding site, and this binding site is specific for the head of myosin to interact with. Allow muscle contraction when there is sliding of actin over myosin. Skeletal muscle contraction appear when there is sliding of actin over the myosin. So again, that is the thick filament, and that is the myosin, and that is the head of myosin, and as we mentioned, it has the capacity to hydrolyze ATP. It has an ATPase enzyme, and this head of myosin is specifically binding with a specific site here on actin called the binding site. And the interaction between the head of myosin and the actin is allowing the process of skeletal muscle contraction by sliding of the actin over myosin. So the actin is a globular protein containing a binding site, and these binding sites are specific for the head of the myosin. We have another proteins. One is called tropomyosin and troponin. They are regulatory proteins. These two proteins are regulatory proteins. They regulate the process of contraction and relaxation. The tropomyosin is considered as a relaxing protein. What's the meaning of a relaxing protein? During relaxation, the tropomyosin prevents the attachment between the head of myosin and the actin binding site. So that is the tropomyosin. So what is the role of the tropomyosin? Tropomyosin cover the binding sites of actin during what? During relaxation. That is the tropomyosin. We have another protein which is the troponin, and troponin is formed of 3 subunits. It is formed of 3 subunits. It contains troponin C, troponin I, and troponin T. What is the troponin? Troponin is formed of 3 subunits, and this troponin is specifically bind with calcium, especially troponin C. It binds with calcium, and when the troponin C binds with calcium, it will produce conformational changes or a change in the shape or the position of the tropomyosin, producing movement of the tropomyosin away from the binding sites, allowing the head of myosin to bind with actin. So when calcium that is stored in the sarcoplasmic reticulum is released, and when it is released, it goes and binds with the troponin C, and this will cause movement of the tropomyosin away from the binding sites, and this will allow the head of myosin to go and form the cross bridges and the start of the skeletal muscle contraction. That is the protein which is known as the regulatory protein. We have tropomyosin and we have troponin. Tropomyosin during relaxation covering the binding sites on actin, preventing the head of myosin from interacting with it. During contraction, calcium binds with troponin C, moving the tropomyosin away from the binding sites, allowing the bending and binding of the head of myosin to the binding sites on actin. So these are what? These are the contractile proteins and regulatory proteins. Myosin and actin considered as contractile proteins. Then we have the regulatory proteins, and these regulatory proteins including tropomyosin and troponin. Lastly, we have the structural proteins. Structural protein include nebulin, titin, alpha-actinin, myosin, and dystrophin. We will start with the first one, which is the titin. What is the action of titin? Titin is the protein which binds the M line to the Z line. It is an elastic protein and can stretch, account for the elasticity of the muscle. And if you look to the titin attaches what? Attaches the M line to the Z line. That is the protein which is called titin. Titin is the protein that attaches the M line to the Z line, and we mention it as a large elastic protein, and it is concerned with the elasticity of the skeletal muscle. Elasticity of the skeletal muscle. Alpha-actinin, it is binding actin filament to the titin. Binding actin filaments to the titin. So titin. So it binds the actin filaments to the titin. That is the alpha-actinin. Then we have nebulin. This nebulin attached the actin to the Z line. attach the actin or the same filament to the Z line. This is called nebulae. Then we have a protein called myomycin, and this myomycin is forming the M line. Myomycin is a protein that forms the M line. Lastly, we have the protein which is called dystrophin. Dystrophin protein, and dystrophin is the protein that bring the muscle tension created by the sarcomere, bring the muscle tension created by the sarcomere and bring it to the sarcolemma, and it brings it to the sarcolemma. So it transmits the force of contraction created by the sliding of the actin over myosin, and it brings this to the sarcolemma of the muscle fiber. Bring it to the sarcolemma of the muscle fiber. So we have the structural protein including titin, alpha-actinin, myomycin, nebulin, and dystrophin. By the end of the lecture, I would like to introduce my thanks, and I wish you the great success in understanding of this lecture. If you have any question regarding to this lecture, please you can contact me through the email. Thank you very much.